Okay, lovely. Thank you, everyone. So I'll do a little quick intro and then I'll pass the floor to you. So it's my pleasure to introduce Professor Adrien Peyrache from McGill University, and many of you know that's where I did my PhD. And his lab, fun fact, was just down the hall from the lab of Nathan, where I did my PhD. And I don't know if you remember, I think one of the first interactions we had, I felt so embarrassed. It was a cold day, and we had a talk, I think, down at the Grand Pré, and I had a kettle of boiling water, and we were in the elevator, and I just dropped the entire kettle on the laptop. And you were in the elevator with me, and I was like, every day since then, since I met you, I was feeling terrible embarrassed. Anyways, thank God you do remember that. Anyway, so many of you know that the lab from Professor Pirash just published a paper in Nature, and the interest of the lab is around spatial navigation and brain dynamics. So they draw on many of the things that are familiar to us, so computational approaches, systems neuroscience, and state-of-the-art EFIS and imaging. And so that's why we're here today to really try and absorb information from you, and hopefully we have an interesting discussion at the end. Before you start, is it okay if we stop you along the way, or do
Yeah,
you want to?
absolutely. Whenever
Okay,
you want. It's fine.
lovely. Okay, the floor is
Cool.
yours. Thank you.
Cool. Thank you. Well, thank you so much for your kind invitation. And I've made the joke for the past week now that I was giving a talk in Australia tonight. So just by Zoom. But no, it's a pleasure. So let me do this. Wait. All right. So what time is it over there?
9 a.m.
9 a.m. Good. All right. So yeah, as so again, thank you for the intro, and thank you so much for the invitation. Please, sorry for the Sydney that actually is broken here. I don't know why. And please do not hesitate to interrupt. I'll provide you like somehow like a vision of like view of like what we're doing in the lab, and I'll focus on a few recent studies published or unpublished, and so especially the unpublished ones, but like comments are more than welcome. For the published one, it's too late, but of course I'm happy to discuss anything. And so some background here is that I'm my background is actually not in biology or neuroscience. My background was in physics and chemistry, and when I start, like in my senior year as a student, I started to read about these weird materials called spin glass that have memory, and this is when I first heard about like some analogies that had been made, especially in the 80s, early 90s, between the systems and memory in the brain, and this special place in the brain called the hippocampus, you'll know about it, that could basically be modeled at some level with the same kind of physics equation than these weird, interesting physical materials. And that was the first time for me that I ever heard about like these places in the brain, and I started to read it and became extremely fascinated with this. And it turns out that the seminal work, as I'm sure you all know, was done by Brenda Milner, who's like still a colleague of mine here. She's 107 years old. I think now she's retired. Actually, it's been a few years she's retired. And describe this patient HM back in the 50s, who had undergone surgery, resection of the mesiotemporal lobe that contained the hippocampus, and what they realized after the surgery. Apparently, there had been like several patients before that, but they were not possible. It was not possible to study them. But like studying this patient, they realized and they really assessed that this patient could not form new memories. Without the hippocampus. Okay, so he was really suffering from anterograde amnesia, but also the recent memories were impaired, meaning that he was suffering from retrograde amnesia, but not entirely like the entire life. Only the most recent memories were impaired. So the idea is that yes, the hippocampus is necessary for the formation of long-term memories, and more into the details here is that the hippocampus will basically encode information from the cortex, like, all the high dimensional sensory and other executive information from the cortex will be encoded somehow in the hippocampus rapidly, and on what has been called a cognitive map by Tolman, for the first who actually coined the term. And again, the idea that emerged also in the 70s from David Marr was one of the first, but other people like Yuri Brzezicki in the 80s, Bruce McNaughton around the same time, the idea emerged that this information that is rapidly encoded in the hippocampus will be replayed then to help the cortex to learn. To learn and to form a long, trace of these memories, because like memories are eventually stored for the long term in the cortex. And one hypothesis that again was first also already formulated by David Marr somehow is that this process should take place during a time when the brain is not engaged in active behavior, don't have to be engaged in sensory processing of the external world, and one ideal state for this will be sleep. Okay, and this is exactly what has been shown in the early 90s. So let me actually show you here. So these are like electrophysiological recordings done in rats, in this case by Albert Lee in Matt Wilson's lab. This is not the first demonstration of the phenomenon, but perhaps one of the best illustration. In rodents especially, even if it's also the case somehow in humans and non-human primates, many of the neurons, principal cells in the hippocampus, code for the location of the animal in its environment. They are called place cells. In this case, we see the place field of 10 different cells, which are coded by different colors. And as the animal is running from left to right on this linear track, naturally, you have a sequence of, a sequential activation of the neurons, trials after trial. So here you see about 10 trials for each cell, and you see this sequential activation of this ensemble of neuron on this 2.53 meter long track, all right? And this somehow will reflect this transformation of cortical activity into this hippocampal code. So for some reason, the hippocampus will encode this high-dimensional cortical activity into a code for space. And what was observed, the phenomenon that was discovered is that during sleep following this task, the same sequences are replayed. This is cool. So sleep replay, you see here the same sequence. So there are two key properties here. One is that, like as you can see, it's highly compressed in time. So this whole sequence that formed, let's say, for a few seconds during wakefulness, now is replayed in about like 100 to 100 milliseconds. And it's associated with this fast oscillation in the hippocampus called ripples, and form a complex called the sharp wave ripples. And now there are like, a band, like a large body of papers showing that these events are instrumental in the formation of long-term memories. For example, you can selectively suppress these events, and the formation of memory is impaired. These events also, is important to note, happens specifically during this phase of sleep called non-rapid eye movement sleep. As you may know, like a sleep cycle is divided first into non-rapid eye movement sleep, then followed by REM sleep, rapid eye movement sleep, and it repeats again and again. Okay, so that's the first part of each sleep cycle in which these events take place. But so basically that was state of the art when I started my own PhD, when I decided to shift to neuroscience after like everything I had read and become, absolutely fascinated with. And I work on this phenomenon and replay and how it interact with the cortex, but I grew some frustration or like some, let's say, I became a bit, conscious about, the reality of the model that everyone was talking about, like meaning that the cortex and the hippocampus will be engaged in this dialogue for the formation of new memories. And one of the problems is that you assume here that you can align somehow this very high dimensional representation, both like the sensory representation. So this is, for example, me seeing the Eiffel Tower in my hometown, Paris, and aligning this high dimensional representation, as you can imagine, like even in the morning or in the afternoon or in overnight. This is not even the same representation from the same point of view. And aligning with this high dimensional representation of space. The hippocampus code for your location in your city, in your environment, but of course it encodes multiple maps. These maps themselves are modulated by non-spatial factors. So even from a pure machine learning perspective, like aligning these representation makes no sense, or will take an infinite amount of time to actually be aligned. And one possible solution, maybe not the only solution, but one of the possible solution is that you have this intermediate representation, especially low dimensional representation, that will allow actually to align this representation in the cortex and the hippocampus. Okay, so this is a point where you ask yourself, is like, okay, so but what do we know about, like, this interface, like the input to the hippocampus? Like, does the hippocampus really receive direct input from the cortex? Well, no, they are intermediate structures. So what do they code for? And these anatomical circuits are very well described, like it's been almost a century now that we know somehow like what is a global architecture here of this system. And one key component of the system is a so-called Papez circuit. And so the hippocampus, the hippocampal formation, which includes the entorhinal cortex, receive input and is mutually connected with the subiculum, okay, which is, let's say, part of the hippocampal formation itself. However, like on the other end of the spectrum, of this circuit, you have the subcortical structure, especially the mammary bodies, which is somehow, if you want, like the limbic hypothalamus, that receive input from the brainstem, like very low level signal from the brainstem. And there's this beautiful architecture here in which the mammary bodies will project, unidirectionally to the anterior thalamus. The anterior thalamus itself will project almost unilaterally to the subiculum, and the subiculum will project back to the mammary bodies. And the activity then will be organized in these loops, okay? Lesion to any of the structures, like the mammary bodies or the anterior thalamus, leads to impairment in memory or spatial navigation that are almost, if not more severe than lesion of the hippocampus itself. Okay, so it's not just the hippocampus. Like integrity of this entire network is absolutely necessary for the emergence of these abilities. So this is like a general circuit here. However, when you look at it more carefully, you realize that actually it's not just one big loop, it's actually multiple loops in parallel, at least three loops in parallel. And in one of these loops, if you put an electrode in a freely moving animal, you're gonna actually find and observe these beautiful cells called head erection cells, which were described in the early 90s by in the lab of Jim Rank. And what you're seeing here on the right hand side is like a typical tuning curve of one of these cells. This is fire rate as a function of head erection of the animal in the horizontal plane, and you see like this cell will fire for a very specific direction of the head. Okay. And I'll let you appreciate the signal to noise ratio, meaning that this cell will fire almost absolutely no spike outside the preferred direction. Okay. And altogether, different cells will code for different direction, and they form like, a system that is akin to a compass for the brain. And I told you this is like one of these sub-circuits. So these are the three structures that are involved in the processing of the head erection signal. And these are, three structures that I'm gonna talk about again and again in this talk. So the subiculum, the part of the subiculum is called the post-subiculum. The mammary bodies is the lateral part, the lateral mammary nucleus, and this is the anterodorsal nucleus of the thalamus, very specifically engaged in the processing of this head erection. And what we know is that this head erection signal emerge from the vestibular system, the semicircular canal, which basically act as like a gyroscope for your head. And a few synapses, they are transformed in the mammary bodies into a head erection signal. So what is the belief is that this is the generator of the signal. All right. And then people assume that the anterior thalamus, as mostly the thalamus in general, is a relay. It takes information and relay this to the cortex, like, dorsal, lateral geniculus nucleus, relay the retinal information from the retina to V1. And the post-sub does actually another job, will be to align this or to encode this representation, sorry, to the external world, right? Because you have a signal that emerged from inertial acceleration of the head, so it lacks a global, anchor. So this is where it will happen, like receive input from the visual system, and this is basically what aligns you to the world. It updates the signal, and then by this feedback, all the system is in register everywhere, aligned to the external world, and updated by the movement of your head. All right. So what I'm gonna show you today in the next slides is like, we will revisit basically every of these 3 points, like the generator, the relay, and the update. Okay. So how do we do that in the lab? So we study the system now is like the kind of technology that allow to record many neurons simultaneously in freely moving animals. So here you see a freely moving mouse. This is one of our favorite system in the lab, even if we also do some rats. In a circular environment with a white card here, which is the Eiffel Tower for a mouse in the lab. So like an anchor to the external world. And we insert like these silicon probes, different types, for example, in this case, a 64-channel probe that record from the entire extent of this post-subiculum or head direction cortex. And, you can then relate the firing rate of each individual cell with the direction of the head, here represented as a polar plot, and you see 4 cells with 4 very specific preferred direction. And of course, we record many of these cells simultaneously. Here you see in purple all the cells that in one recording only were categorized as head direction cells. All right, and you see that they all have different direction, meaning that we have access to all the information encoded by the system somehow. Why that? What do we do with this? Well, one way to represent this classically is a raster plot. So here you see the activity in the population of 86 cells in time as the animal is awake. And so each row here is the one cell, each tick is one action potential. And as you can see, like by eye, you don't need any sophisticated, mathematical tool. You see the activity in the system that moves as the animal move. Here we have in black the actual head direction. So this is what we call the activity packet in the population that moves as the animal is turning the head. Just imagine this is like a needle on your compass that moves, whenever you're turning the head. It's amazing. You can even put it on a loudspeaker if you want, like one of these cells, and you put your mouse like, on your hand. I've done it multiple times. And what you will really hear is like nothing, like bzz, bzz, and each cell will fire for a very specific direction like that. But you can still, use some of these tools like a Bayesian decoder, for example, that will then transform the activity back into an angle, showing how reliable the decoding is compared to the actual head direction we're tracking with a camera. All right. So now that we have access not to single cell activity, but to population activity, what can we do with that? And there's actually many things we can do. The first one is to try to understand like how this activity is organized at the population level. Okay, this is what really now is what is possible with this kind of recordings. And one fascinating thing to me still is to show like that the activity in this structure is highly organized and internally organized. Let me tell you how and why. If you slice the activity, in population vectors, okay, so you count the number of spikes from each cell, like in each of these vectors, you give it to some, dimensionality reduction technique, not knowing anything about the behavior or the tuning curves. PCA works okay, but ISOMAP, UMAP, all these tools, and you ask these algorithms to plot it on a 2D map, naturally it will show you a ring. So now each point here is a population vector and the color code for the head direction of the animal at the time the population vector was recorded. And you can see it's somehow expected, but it's still beautiful to see, at least to me, that naturally here the organization of the population activity is a ring because this is a network that codes for an angle. Okay, and there's nothing in the middle, meaning the activity never explodes this place here, and there's no activity far from the center. It's really restricted on a ring, coding for an angle. So somehow, if you want, this is what is called usually a low-dimensional ring attractor. It will correspond to some it would correspond to a network that visit like a low energy, whatever energy is in this case, a low energy landscape that will have the shape of a ring. Okay, and there are like all these states that are forbidden of high energy that the system barely like never visit. All right. So is it like something that is really internally generated, or is it something that is, influenced by the external world, by vision, and by other senses? The way, like one of the way we tested for it is, as done with replay, is to record from the same cells now, but during sleep. So it's pretty easy to achieve. You put the animal back in its home cage, and the animals will fall asleep, and you record the same neurons. And what we realized is that the activity was still perfectly organized in this network during sleep. And there were a few more properties that were fascinating. During rapid eye movement sleep, okay, which is virtually indistinguishable from wakefulness, the system behaves exactly as if the animal is awake. So it's like if I give you the data from REM sleep, it is virtually impossible to tell that the animal is asleep. You see the head erection cells that, first of all, they are still perfectly organized, meaning there's one activity packet every time, at any time, and it moves at the same speed as wakefulness on average. All right, so interestingly, this is the sleep phase in humans where we have the most, like, we're dreaming the most. So somehow it shows that if we extrapolate that we dream in real time, right? Whatever is happening in our dream takes as much time as it will in real life. But anyway, and the second thing I wanted to say is that if you now also do this isomap kind of analysis during REM, you see that you also have a ring. So the ring is something that is internally generated. It's like, governed by properties of the network, of single cell, many different things that really constrain the activity on this ring. However, as I told you a few slides before, what I am really interested in is this non-REM sleep phase where memories are replayed in the hippocampus. And if you look at non-REM here, looks noisier, all right? However, if you zoom in, you realize that it's actually still very well organized. There's still always one activity packet. For example, here it does like a very fast sweep, and then you see this packet that somehow jumps from different direction, but at any time there's only one and only one angle that is encoded. Okay? So still very well organized, just with a different, with a different dynamic and different kind of behavior of your needle. So the question we had when we made these observations, like, wow, this is amazing. This is like the same organization during sleep, before or after the task. And it really raised the possibility that this system was hardwired. I was like maybe it's always the same. So to address this question, the problem is that we can't really use electrophysiology as I showed you here. You need to change technology if you want to track the activity of the same neurons now across multiple days. Okay, you can potentially do it with electrophysiology, but it's always a bit harder to assess. So there is one technology that allows you to do this is calcium imaging. Uh, Here in this case, what you do is like you transfect neuron with a calcium indicator called GCAMP, in this case GCAM6, that basically will serve as a proxy for neuronal activity. You insert a green lens, which is basically like a window into the brain, and you put a microscope on top of the animal, like a miniaturized microscope that is like only 2 grams. The animal can perfectly move in space with it on the head, and that will image, like really take movies of the activity at the bottom of this green lens. And here you see an example of the fluorescence, in green for the different head erection cells that were recorded. And again, you see this activity packet moving, in the population as the animal is moving around in the environment. All right, and you have exactly the same data as with electrophysiology, more or less. So again, our question was like, is it hardwired? Like, is it stable? So the first thing we did is pretty simple. So we actually let the animal explore the circular arena I showed you before every single day for 4 weeks. I mean, every weekdays, I let Sophia, the PhD student, to work on this to take her weekend off. I'm not this kind of PI, and but like for 20 days. And as you can see here, this is the average fluorescent taken for 4 different days. And you see this like so-called region of interest, meaning like the spatial footprint of different neurons in the population. And you see that we have exactly the same field of view for like across 4 different weeks. So we are capable of isolating, individual neurons for all this time. And what we realize, like for this cell, for example, here in red, the tuning curve is exactly the same. It doesn't move. It doesn't change. Okay, you have like this perfect representation of stability in the system for one environment and across all the animals were recorded in exactly the same protocol. So it seems to be hardwired. All right, so here we have stability at 2 levels, at least in this case. We see that we have stability of the internal organization of the system somehow, and to the way it's anchored to the external world. So let me show you a second example. We did a lot of different experiments here. I'm just showing you like the surface. We did another example, like another test is like, is it the case across different environments? Do we have remapping across different environments? So we then recorded, from the same neurons again, but now 2 weeks in 3 other environments. Okay, starting from the circular, but then different geometries, different size. And we show here like 2 cells that code for 180 degrees apart, like opposite direction. And what you realize is that in each environment they remap, relative to the proximal cues in the environment. However, they always fire 180 degrees apart. And I show you here just like an example pair, but just imagine that actually, believe me, or check the paper, it's the case for the entire population. You have in conclusion, you have this very rigid, ring population coding for head direction. And in a novel environment, like in a new environment, it will acquire like an absolute orientation and then maintain this absolute orientation for like as long as the animal explores the environment. So we tested it like across several weeks, and it really seemed that first this orientation is acquired like very rapidly at most, like, at maximum 20 minutes. This is like how long the animal were allowed to explore like the environment for the first time. But we've had some data in the lab showing that maybe as little as 5 minutes is sufficient for this orientation to appear. And it's stable for like up to 4, if not 6 weeks. Okay. After 4 weeks, you start seeing like some drift in the absolute orientation, but for 4 weeks, just one visit on day one, 4 weeks later, with no visit in between, you put the animal back in the same environment and it goes same orientation. So it's 4 weeks in the life of a mouse, which is like, I guess, many years in human life. So that, you visit a place for the first time, you establish like very quickly this mapping between the external world and your internal representation of your orientation, and this mapping then is very likely very stable for a very long time. Okay, so meaning that once you go back to the same place, even months later, you don't really have to establish this mapping once again. It's already there. You feel immediately oriented, and you know like where to go, and you know how to navigate this environment. Okay. So let me summarize like, this kind of introduction here. So the head direction system, it's a simple, let's say, put it this way, low dimensional attractor that encodes head direction, meaning that it's an internal ring attractor on which you map, the direction of the head during wakefulness. It's a signal that remains coherent during sleep, and we'll see here. Sorry, I shifted the slide, but we'll see how it's related to hippocampal activity, and this attractive dynamics is maintain a stable orientation memory over weeks. All right. So I wanted to show you like another thing that is published before we move on to some unpublished data and going back to sleep afterwards. I told you in the introduction that this is like in the post-subiculum somehow that the head direction signal would be anchored to the external world. And this belief is based on lesion and on many other studies. However, like exactly how it works is still very unclear. Like how do we detect landmarks in the environment that could be relied upon to orient ourselves? All right. So I'm not saying that we've now understood it fully, but we have some new data that gives us some like new leads in terms of like what is going on. And actually, I really like this study because it really started like on a totally different subject, with totally different people until like we connected the dots. So it started actually with a colleague of mine, Stuart Trenholm, with whom I actually collaborate on totally other things, head direction, but in blind animals and other things I'm not talking about today. But he was working at the time with a colleague, someone in Germany, Emily Massé, who actually turns out that actually we have the same alma mater and knew her like I had known her for a long time, and together they were actually asking the question, where in the brain objects are encoded. Okay, so Stuart is like someone coming from the field of visual neuroscience, and this is like one of the hot questions in visual neuroscience, like where are objects encoded in the brain? So for that, they use a technique that Emily developed during her own PhD in Vivo, which is functional ultrasound. It's if you never heard about it, it's like say it's more or less the same as fMRI, but at a much more precise spatial temporal resolution. So it's still a signal of bold. Okay, so you see like increase in blood flow, but at a resolution of about 50 micron, and you have it like almost in real time. So even if it's a bold response, which is slow, you can actually read out from the brain at a very fast rate. So they had fixed animals, and they showed the animal objects and random objects. It could be a table, a wheel, a dishwasher, a plane. Okay, and a scrambled version of these objects. Okay, so this scrambled version preserve a lot of the same properties, but they lose, they lose the fine spatial arrangement of all the components. They preserve the contrast, they preserve like many the texture. But so it moved from an object to a texture like, kind of stimulus. And the first thing you see when you do like, whole brain assessment of the activation here is that whatever you present to the animal, you're gonna have this explosion of signal, of both signal in the visual system, V1, V2, and the superior colliculus in these figures that are quite expected. However, now, if you ask the question, which place in the brain you see a statistical difference between an object and the scrambled version of that object, surprisingly, it's a head erection system. So this is something they observe, like after recording from a few animals, like in this task. And one day Stuart knocks on my door right here that you see behind me, say, like, can we talk? And of course, then we understand that this is something very important and interesting. And I put immediately two people on this to understand better. So you see here again in red, these are the places in the brain significantly modulated by the object, and it was like very precisely the post-sub. I showed you before. A bit of the retrosplenial cortex also, which is known to be involved in the process of head erection. So what we did then, we repeated the same experiment with Sophia again and another great PhD student in the lab, Henry Denny. But now we actually coupled these recordings like during object presentation. We did it with electrophysiology, and the same animal, we did some freely moving behavior. So the animal explored again an environment. We had head erection cell. Here you see two of them. And then, the same animal right after was head fixed and objects were presented. And one thing we observed is that some of the cells were activated by objects. Okay, while other cells were inhibited. And we were like at first, okay, classic, you see an increase in bold response. Doesn't necessarily mean that all the cells in the system are activated. It's more complicated. So maybe there's something going on. There's clearly a code for objects, but it's more complicated. Until we actually realize one thing is that all the cells that were activated by objects fired for the same direction when the animal was freely moving. Okay, so here you see different cells recorded during in this session, and their preferred direction is represented on a circle. And you see that all the cells that had a positive visual response to object were all firing for, let's say, this west direction in the environment. All the cells that had negative response coded for all the other direction. Okay. So what we believe at this point is that, and we saw it for all the mice, sorry. And so what happened here is that this direction here, like that direction, is actually the direction of the head of the animal in the head fixed apparatus. Okay. So it's unrelated to like, it's random compared to the direction here in the open of field. But of course, when your head fixed, your head direction system still code for head direction. And then we understood that it was actually that direction coded by these cells. So these cells are active because the animal is head fixed for this direction. They show an object, and like the cells that are already active are even more active. But the cells that are coded for other direction, they are like firing at low firing rate, let's say. But now that when you present an object, they are even more hyperpolarized. Okay, so you increase the gain in the system. You make the cells that are active more active, you make the cells that are hyperpolarized even more hyperpolarized. So this is head fix. So head fix has a lot of drawbacks, right? Like the animal cannot move the head. So is it the case in freely moving animals? So we actually run a few experiments, but including this one, which I think is the most, striking, is that the animal were put back in the environment for freely moving behavior, but now we literally printed this object on a piece of paper and put the object on one end of like on one of the wall, on one side of the wall, and the scrambled version of this object on the other direction. And then we looked at whenever the animal was facing the object versus facing the scrambled version of the object. And as expected, we see that head direction cells increased at firing when the animal was looking at the object, but not when it was looking at the scrambled version of that object. So there was something very specific happening whenever the animal was seeing an object, even if it just printed, a black and white on a piece of paper. So to summarize this, can show it with the schematics, is that at any time you have the head direction signal, all right, but with some background noise if you're a bit lost. But suddenly there is this very, how to say, well identifiable landmark in the environment. Like if you see a forest, you cannot use a forest to orient yourself. But if there's an isolated tree, that is a good landmark. All right. If you're in a prairie, it's very hard, but suddenly you have the moon, and the moon will be the object you want to use to orient yourself. And suddenly this confidence in the encoding of your head direction is literally, how to say, leads to the boost in the gain of the system. You increase the firing of the cell that are activated and you decrease the other one. Okay. And one possibility here, which is interesting, even if it's, still like an hypothesis, obviously, is that this object detection in the environment may have evolved for spatial orientation, first and foremost. And then, primates like us, like, sure, we see like a chair, we want to sit on it, we see a mug, we want to drink coffee, and like objects are part of our world and of our affordances, but maybe like at first and foremost, like this detection of things around us was really for spatial navigation and for spatial orientation. Okay? So, this is all I wanted to say about wakefulness. I want to go back to sleep now, and tell you what we believe is happening in the system and how it relates to to memory formation. And as a transition to sleep, I want to show you what is happening at the transition from wakefulness to sleep. Here we recorded, cells in the thalamus, head direction cells, as the animal is in its home cage and about to fall asleep. Just imagine you like going to bed, finding a position, a comfortable position to fall asleep. And you see here the actual head direction, the animal moves one last time. And in red, you see the head direction that we decode from this population of cells. And you see that we have a perfect tracking at first, but suddenly when the actual head direction kind of stop moving somehow, you see that the decoded head directions cell actually becomes noisier. Okay? And I'm gonna skip this. I'm gonna show you this immediately. So this is a cognitive space, like this isomap I showed you before. Okay, so this is your population from wakefulness. And we can now represent what is going on in the data. as a trajectory on this space. So you see here, and this is where the animal is at this point, at the beginning of this orange rectangle, and he's like turning the head about 180 degrees, and we see this trajectory, so like the needle moving in the population. All right. But now let's look at this moment there. Okay, so now this is for the first time. So this is the actual head direction of the animal. The animal is not moving. And what we see in the data for the very first time, now the head direction cell, they do a full sweep of the head when you project this. And I'm sure you've all experienced this. You're falling asleep and suddenly you have like a full sweep of the head. And it's funny to look, it's quite cute because you see that there is this full sweep here. And when you look at the video, you see that the animal like will wake up like this, a bit surprised by what just happened, and will fall back asleep. It happens again, then wakes up, and you have this whole, the so-called sleep onset period here until then the animal falls totally asleep. So you see, like, first the animal is totally mobile here. The system now, the head erection is totally disconnected from the external world, and we have EEG markers to really show very reliably that this is sleep. But this whole transition here of how the system actually transitioned from this sensory engaged to sensory disengaged states. And it's super interesting. Long story short, it really looks like a phase transition. I told you my background is in physics, and when you see actually in the data, it seems that it's like a type 1. It's like either engage or disengage, but there's nothing in between. This is not a smooth transition. It's an abrupt transition between these two possible states. So this is what we see here. These cells were recording in the thalamus. So we see this in the thalamus. Then it raises the question, it's like, but where does it start? I remind you, the signal emerge through like signals from the vestibular system. So is it the entire head direction system that start generating these fast sweeps and disconnect from the external world, or is it like at a specific point in the system that the signal emerge during sleep? Okay, so this is like a question that Guillaume Viejo, one of the first postdocs in the lab, worked on for many years with the help of Sophia again. And this is a paper that is currently under review and available on bioRxiv. So where does it start in the system here? So to answer this question, as a young PI, I discovered that it was super nice that I didn't have to do the experiments anymore, and I could just ask people to do some very complicated things. And I can't actually believe that they managed to do this because I was like, you know what? I've published as a postdoc, the organization of head erection cell here in the thalamus during sleep. So why don't you actually record one synapse before in the mammary bodies? But then you have to realize how complicated this is because we're talking about a nucleus which is about 100 to 200 micron in width, 6 millimeters deep in the brain of a mouse. Okay, and you want to target this with like a silicon probe. So you have to be perfectly aligned. You have to get your coordinates perfectly fine. And even though then you want to do freely moving behavior, all kind of crazy stuff. And not only they managed to do it, but they even managed to do in many animals the recording of these two structures simultaneously. So now we're talking about two silicon probes in a freely moving mouse. Okay, or a free moving mice, cohort of mice. So let me show you what is happening. So here you have your tuning curves from thalamus and mammillary bodies. And interestingly, and this is a well-known fact already in the field, is that the tuning curves are slightly larger in the mammillary bodies. During wakefulness, unsurprisingly, you see this activity packet as the animal is moving around like that we know, and we see it here also in the mammillary bodies. You see that it's a bit larger. It's just because the tuning curves themselves are larger. Okay. During REM sleep, I'm not gonna show you this. It's exactly the same. Again, it's virtually indistinguishable from wakefulness. So during REM sleep, and I'm happy to discuss this if you're interested, the signal emerged somewhere we don't know, actually, like far away in the brainstem. However, during non-REM sleep, we saw something very interesting, is that you see the signal, again, in the thalamus, still perfectly organized, sweep, sometimes jumping, but at any time there's only one direction that is encoded. But in the mammillary bodies, it looks pretty random. So you lose this coordination, okay? And at this point, it tells you two things. First, is that the thalamus is clearly not just a relay. The thalamus is capable somehow to take perhaps a noisy input. Let's assume there's still some coordination. It takes a noisy input and it transforms it into a beautiful, nice signal. Okay. The second possibility is that actually this activity is totally disorganized and the thalamus has its own generator. It has its own attractor. Okay. And it's revealed now by looking at the system during sleep. Even if the attractor will still be there during wakefulness to filter out any kind of incoming noise. So let me show you this in another way. So these are like, awake and non-REM sleep, raster plot, and we can represent these this data again by projecting this with some tools. Like here, it's like a so-called kernel PCA. And you see that from element to 80 as the animal is awake, it's one ring transforming into another ring. So the thalamus doesn't have to do really anything here. It takes a ring, relay the ring. But during non-REM sleep, now it's not a ring, it's a disk. It's not totally disorganized. There's more red here, more blue there, but clearly there's a lot of points that should not be where where they should not be. Okay, so it's not a ring anymore. But the thalamus somehow is capable of transforming this into a ring. Okay, so these data are recorded simultaneously. Okay, so here this is not fake, this is actual data, and you see what the thalamus is capable of doing. All right, so you can quantify this. I would call it population coherence. It's pretty simple. It's actually the correlation of the pairwise correlation between wake and sleep. And you see that in AD, the coherence so one is perfect coherence, meaning it's exactly the same structure as wakefulness. So in the thalamus, you have coherence that is almost the same as wakefulness, okay, one. But in the mammary bodies, it drops. It's not zero, but it drops, meaning that there is still some coordination there, okay? So the question is like, again, as I said before, is it the thalamus that is capable of taking this noisy signal and extract the ring, or is it the thalamus that actually generates the signal? And let's not forget one thing is that the mammary bodies itself receive feedback from the subiculum, from the cortex. Okay? So one possibility is that whatever remains coordinated in the mammary bodies is actually inherited from the cortical feedback. Okay? So we tested this directly, and this now we have all the tools to actually do this. So what we decided here to do is to switch off entirely the post-subiculum with optogenetics. To do that, like the best technique, it's now well known, you activate the inhibitory neurons. So you use these Cre lines, these mouse lines that express Cre. You can then inject a virus, you express this activating opsin called channeled opsin in all the inhibitory neurons, and when you switch on the light, boom, the activity in the network is gone, especially of the excitatory cell, the head erection cells. So you do this optogenetic manipulation while recording in the mammary bodies. And look at this example here, is that here you see this is a moment, again, non-REM sleep in the mammary bodies, in which you still have somehow some organization. So it's not totally disorganized at this moment. But you switch on the light and the activity becomes totally noisy. You lose all coordination. Okay, it becomes like random spiking. You switch off the light and something weird happens, some rebound, not super organized, and maybe then it will come back a few seconds later. So now you can compute the population coherence, and you see that with this optogenetic tool, when you switch on the light, now the coherence is zero. There's nothing left. So it means that the mammary bodies, without feedback from the cortex, is incapable of generating a signal during non-REMs. Okay? So what about the thalamus then? What does it do without its cortical, partner? We did exactly the same optogenetic manipulation, but now we're recording the thalamus. And you switch on the light, and it's as if nothing happened. The thalamus doesn't care. Continues to generate a beautiful signal, and that moves around, okay? And you see the coherence remain exactly the same as the control. All right? So, long story short here, during non-REM sleep, the head erection signal is generated very likely in the thalamus. Okay, so forget about the textbook idea that the thalamus is just a relay. The thalamus is actually a beautiful structure that is endowed with its own dynamics, its own generative capacities. Okay, and we see it with the head erection. perhaps, we'll see in the future that it's the same for the visual thalamus, for the somatosensory thalamus, whatever. But clearly, there's a lot going on there. The signal was generated here, and then will be conveyed to the post-subiculum and certainly to the hippocampus and trianal cortex. And there's another part of the lab now trying to understand what is going on. You have the signal that is generated there in the forebrain, independent of what is happening in the brainstem. How does it play any role in memory formation? Let me show you one example. It's a paper that was published a few years ago now already. We're actively working on the lab in the lab on this question now, but some projects take longer than planned. Some data are just being recorded, but basically the idea will be the same. So again, we record head erection cells in the thalamus. That's your isomap. You know this already. And here, the only thing we added to this project is that we had the LFP or EEG, if you want, in CA1. Okay, so in this part of the hippocampus where we have ripples and replay. And you see here, this is the LFP, and you see this fast oscillation. This is your moment of replay, the ripples. Okay, and as I showed you before, what we can do now is to project this raster plot, now you know them, onto this cognitive space, okay, as a trajectory. And if we do it, sorry, the forbidden state, like I don't want to talk about it. Too much. And you see here that, well, we start from this moment of low activity. It doesn't really matter why at this point. And you see the activity goes on the outer ring. Okay, so again, in color, you see the activity during wakefulness. In gray, this is during sleep. So you see that you go at same activity level as during wakefulness, all right, because the radius here, the gain is basically the firing rate of your population. 0 is around here. So you have the same firing rate somehow as wakefulness. And I let you appreciate how the angular head velocity stops. It's like you create like the needle grows in length, stops moving, and you have a ripple in the hippocampus. And right after the ripples, it moves somewhere else. And we see it over and over and over, So you start from some place, you go on the outer ring, you stop, and you have replay in the hippocampus, and you move somewhere else. And you see it here across animals, you have increasing gain right before ripples or sharp wave ripple in the hippocampus. You see it peaks right before, and you have a drop in the angular head velocity of this signal. Okay, as if the head erection cells, again generated in the thalamus, will pick a direction at random, stop, and then let the hippocampus generate a trajectory along this direction. And now we have data showing that it is exactly the case, that yes, hippocampal replay occur along this direction that are generated in the thalamus. And we also have computational work showing also exactly the same thing, is that a recurrent neural net train on different things that in which a cognitive map like looking like the hippocampus emerge during sleep. If you drive it with somehow like a direction signal, the same thing will happen. Okay, so it's another paper on bioarchive right now where we've really looked at these kind of processes from a computational point of view. So let me finish by coming back to the original question, like what will be really the role here of the system in memory formation? And I told you, like, this somehow impossibility of aligning these cortex and hippocampus directly. Even if they're engaged in a dialogue, they cannot be engaged in a direct dialogue with each other. It would not make any sense. And one of the possibilities is that they have these intermediate structures or features, low dimensional features along which you can align, these representation. The head erection system will be one of these low dimensional features. There may be other. The grid cells in the entorhinal cortex, very likely, at least in my view, will play more or less the same role, but in another kind of dimension. This is your post-subiculum, which anatomically also is actually an interface between the visual system and the hippocampus. We receive direct input from the CA1, but also from the visual system, and vice versa. The controller here will be your thalamus, choosing the random state and setting the state of this intermediate structure. So during sleep, there will be moment of high gain. The system will start, pick a direction, and that will be a query for the system. So it will start in the thalamus, will go to the subiculum until the hippocampus, and the hippocampus will be queried, with a given direction. Along this direction, the hippocampus will then replay a trajectory. You see, for example, just imagine the animal had explored like a square environment. There's a replay by hippocampal neurons. And from this replay, you can decode a direction, again, like at the output of the hippocampus back to the post-subiculum, let's say in this case, okay? And this is very interesting. Even, of course, all this is still an hypothesis, but this is what we're currently investigating in the lab. The interesting thing here is that now you can compare the query with what is, generated in the hippocampus, and it gives you an error signal. You can compare actually the generative capacity of the hippocampus with your query. If it doesn't match, it means that actually you don't have a good decoder from hippocampal activity and that your representation are not aligned. If it matches, say, oh, maybe actually the system is known and the memory already exists. I don't need to replay this memory. But importantly, you have an error signal that you can track and somehow use it to actually update, all kind of synapses. And in our view, that will be what is happening for memory formation, spatial memory formation, especially, or episodic memories during sleep, one of the processes. So this is what we're exploring right now in the lab. All right. So let me summarize all this. So the head direction signal remains organized across brain state and remains stable for months, at least in mice. Perhaps even for the entire lifespan. We have like one animal that we kept for a very long time, and we saw something also that it was very stable. Landmarks boost the animal's confidence in its sense of direction, and that will be this, process through which information is extracted from vision to orient, like ourselves in space. And during non-REM sleep, when memory replay takes place in the hippocampus, the head direction signal emerge in the thalamus. All right. Before finishing, let me tell you that all the data I've presented to you are analyzed with this toolbox that we developed in the lab called Pineapple. Guillaume, actually the postdoc I told you about, developed this while he was a postdoc in the lab. Now he works at the Simons Foundation in New York to actually maintain this package and develop it even further. So it's a very active. They organize like satellite, events and workshop and major conferences like SFN and FENS. Also in Europe, I don't know if they ever gonna go to Australia, but it's free for whoever is applying. 5-star a hotel all paid by Simon. So, I strongly encourage any of you to attend. They never invited me, but I don't take it personally. Let me thank also the people in the lab working on many different subjects, not only Sophia, but many other PhD students and postdocs, all the funders. Then most of the data that are published, actually all the data published by the lab are fully available on the archive if you're interested. Thank you very much for your attention. I'll be happy to take any question.
Yeah, that was such a tour de force. Wow. Fantastic. Thank you so much. Do we have any questions, burning questions in the audience? I can't see you. Oh.
I could ask one.
Yeah,
Um,
go for it, Max.
Man, that was fantastic.
Yeah.
I feel like I could chat to you for hours and just listen to you describe these beautiful data that you collect, and I love I think it's just a beautiful example for students to see kind of how far you can get by just looking at your data and trying to see what it shows you, and then trying to kind of unpack how it works, like ask curious questions about it, and then use the techniques that we have to kind of investigate what could it be. I think that was just such a lovely example in multiple fronts. So my question, I have many, but one that I'm really sort of curious about is whether you've tried to sort of test the kind of neuromodulatory sort of landscape of this sort of effect that you've seen in this circuit. And obviously between non-REM and REM, there's very big differences and in wake, but you could potentially jump in with different types of crelines or different pharmacological blockers, grab sensors and things, and sort of test whether or not some of these neuromodulators like acetylcholine or noradrenaline or serotonin or something might be playing a gating role in changing the likelihood of one of these different attractors to form. So I was just curious if you've kind of chased that down or if you have any thoughts in that space.
Well, thank you for your kind words first, and for the question because you're absolutely right. So one thing we could say is the following: it's because the signal is generated during exactly the same way during REM sleep as during wakefulness. Clearly, the culprit here is acetylcholine, okay? Because acetylcholine is a neuromodulator that is firing like crazy during REM sleep, and no other neuromodulators is like that. So clearly, acetylcholine plays a role. There is like a lot of connection from our input from cholinergic fibers, like the subcortical pathway to these nuclei, the mammary bud, like in the mammary nucleus, but actually one synapse before, especially in the brainstem. So yes, this is something we're really interested in. Too many questions. We're not investigating this right now, but there's a new postdoc coming like in the fall, and I hope this is what is gonna be working on. So the idea, I can give you like another project. It's I guess kind of obvious. Will be like, can you create non-REM-like states during wakefulness or REM by switching off cholinergic input, or vice versa? And during non-REM, can you suddenly create like a local REM-like state, but local just to the head erection system? And how would this be interpreted then by, the rest of the hippocampal formation, for example? So that will be one of the manipulation to do. I hope we'll do it soon.
Yeah,
Yeah.
awesome. Yeah, thanks very much.
Thank you.
Clear?
You're muted, yeah.
Thanks. Oh, yeah, I was trying to lower my hand and mute at the same time, and it was too much for me, clearly. That was such a cool talk. Thank you so much. Really, really amazing. Yeah, I followed some pockets of your work over the years, and to see it all together was so impressive. And I'm really excited about this stuff that hasn't been published yet. It's super cool. Yeah, one thing it just got me thinking about these sort of, I guess, intrinsic signals generated in the thalamus. We've thought a little bit about in another project about sleep spindles and this sort of generative rhythm, which I think has been kind of narrowed down to sort of the thalamic reticular nucleus and this intrinsic, and I think with some cortical input. And I know spindles aren't necessarily a driver of sharp-wave ripples, but they're certainly a thing that kind of constrains them. And I guess I was just thinking how your findings fit in the kind of scale of these hierarchical nested rhythms, and whether that gives you some clues of how to think a bit more about what the thalamus is doing here.
Yeah, no, that's a great question. It's like, how do we, basically reconcile all this with like what we know about non-REM sleep dynamics? So spindles, you're absolutely right. They're generated in the thalamus, especially in the thalamic reticular nucleus, the inhibitory cells of the thalamus. So here there's also like a lot, to me, like some confusion in the field about like how really nested these oscillations are. It is not entirely true that ripples are nested within spindles. Like what is really clear in the data is that the ripples tend to occur at the onset of spindles.
Yeah.
And it's like and what we're investigating actually in the lab now is that it seems that the ripples travel back. I showed you in this query, like they travel back in the subiculum, and actually they will go back to the thalamus and activate the thalamic reticular nucleus. And this is how they will actually potentially trigger spindles. Okay. Interestingly, we looked at it. It's one panel in one of the papers. It's a small panel, and that no one ever noticed. But there is a relationship between the power in the spindle band and how fast actually the head erection moves during sleep. And the moment when you have this very fast sweep of the head erection signal is when you have spindles. What does it mean? I don't know. But it means that when you have what I like about it is that spindle may actually somehow protect your sleep. It protects the sleeper. You hear a click. If it's not something important, you have spindles in your brain. Like you have a K complex, which is basically a slow wave plus spindles. It protects the sleeper that statistically, if you have a click, there might be another click, there might be another note. You need to protect the sleeper from not waking up. And the head erection cell, like you see that it becomes totally disengaged, disconnected from the sensory input. It moves like crazy. And this is somehow also related to what I showed like in just one slide. It's an amazing way that we're thinking of right now to track how much you're engaged. Like even for this during sleep, we see these phases where suddenly the head direction code for the direction of the sleeper. It's like this micro arousal, and then suddenly it becomes disconnected again. So you can track these things. It's an amazing actually way of monitoring, how much you're engaged or not with the sensory input. So during spinal we see this. Um, yeah, so we don't know much more than this. It's complicated, all these are like, feedback loop and I wish I could tell you more, but we're trying these recordings right now. It's interesting. I had a long meeting with one of my trainees this afternoon right on this. Like we're trying right now to do recording in the hippocampus in the ADNs that we've done, but at the TRN at the same time, like to
Yeah,
have
yeah,
like
obviously.
this triple circuit because the cortical feedback, to the thalamus, especially to these primary thalamic nuclei, they mostly go to the TRN, to the inhibitory cells. And we see it's one thing, though, also that we've published is that after a sharp wave, we see a massive decrease in the activity of head direction cells. So it's basically, we show that indirectly that, yeah, ripples, they activate the TRN and that inhibits the thalamus. And this inhibition then may actually trigger by rebound like a spindle. So how then all this like fit together? What does it mean for memories? Oh, will take a long time, but we'll
You don't have to answer that now, but
no, I have no answer. But these
one
are
quick,
the different leads, right? We're
yeah,
exploring. Yeah.
one quick follow-up. I guess if you're teasing apart this kind of, vigilant versus non-vigilant sleep, when your postdoc starts investigating neuromodulatory systems, I guess noradrenaline will be a really cool one to look at, right?
So yes, yeah, absolutely. And this is something like either we monitor it directly or we're thinking of monitoring with, pupil dilatation,
Yeah.
which is a proxy for noradrenaline. And one thing I like, and I just, again, it's very interesting your question because I had two meetings this afternoon. One was on spinal, the first one. The second one was on exactly this, where like what I showed you like on this like with a student with Yushao like this disconnection at the sleep onset. What we show now is that we didn't have recording of the pupil at the time. We just have the system now. But what we used the EMG.
Yeah.
It's easy to extract actually from the recordings. Like it's like correlation in high frequency band in your signal. This is like a proxy for EMG. And we see now, like if we renormalize EMG across animals, we basically see, like we have a curve which is nonlinear, which is really like, kind of how to say a tangent, like how heavy side if you want function, all or none. Like when you have the EMG start high, you're awake, and the signal is highly, it's coding for the current head direction. But as the EMG decrease, there is a threshold in which now the head direction cell suddenly, disconnect. So in terms, like I was telling you, we're thinking about it as a phase transition. So yes, clearly this EMG corresponds to some level, maybe acetylcholine, maybe noradrenaline, something. But think about it as a temperature, for example, in the physics. And this is like, you have a state like ice, you increase the temperature, suddenly it becomes liquid. And for the head direction cell, we see exactly the same. It's like it's rigid, it's encoding your head direction, but now you decrease the temperature and suddenly, or the opposite if you want, and suddenly it becomes like super liquid.
Mm.
Just to give you like an idea of how we're thinking about it now. So yes, we'll then basically try to connect this to neuromodulators directly.
It's super cool. Thank you so much.
Can
I have
I
a
jump?
question. Oh,
Oh, sorry,
sorry, Mac.
Julia. Oh, no, go ahead.
Um, okay, thank you. I had a question about when you were showing us the Tappet circuit. One of the things I immediately thought about was motivation and, reward signals. And so my question is, have you thought about playing with motivation with your animals and, showing competing cues and how that would play out in your studies?
So just to make sure I understand, motivation to do what exactly in this case?
Yeah, good question. So what I was thinking is what if there's maybe two competing locations that the mouse can navigate, and one you make the mouse more motivated to go towards that direction versus, another
Yeah.
direction.
So that task will go slightly beyond the kind of things we do usually, because here will be like competition between two spatial memories, for example. And yes, I'm sure there are super interesting things to look at in this case. But let me tell you what, like in our simple system, what we do when it comes to motivation and reward. There's a whole paper we published, a couple of papers in which we looked and investigated the head direction signal in blind animals with Stuart Trenholm. Because I was like, this is something we got interested in at first because we were like, it's supposed to be vision that anchored the signal. What about blind animals? It turns out that blind animals have perfect head direction signal. They don't see anything that we've controlled for. And what we discovered is that they use olfaction, unlike wild-type animals. So they learn to use olfaction to orient themselves in space. And then you can ask the question, how do they use olfaction? How can you orient yourself with olfaction? They actually use stereo olfaction. So the gradient of odor between the two nostrils is enough for animals, and actually for humans as well, to orient yourself in space. You can learn this, okay? An animal, well, anyway, long story short. How does it relate to your question? It's like, to test for this, we actually rewarded the animal for a very specific direction in the environment. So the animal had to learn to orient themselves like in plus minus 20 degrees. And we used like an internal reward, like artificial reward for that. We stimulated the medial from fiber bundle, MFP. It works beautifully. And the animal, even the blind animals, when there was an otter, they were like capable of finding their correct orientation of their head in the environment, like wherever like we were dropping them in the environment. You block one nostril, they couldn't do it.
Wow.
Yeah.
That's insane.
So that and I have to admit here, like full credit to Stuart, who came up with this idea one day in my office, and I was like, "You're crazy! It will never work," and it worked beautifully. And so now we're thinking of using this reward, task to test like specifically and directly the sense of orientation, the sense of direction. Yeah.
Thank you. Wow. Mac.
My question was coming back to the states of matter analogy, which I think is really prescient and is something that we've thought about quite a bit in-house, playing around with both spiking models of the thalamocortical system and then kind of mean field models. And so I was curious if you'd try to interpret some of the results of your work in terms of thinking about criticality. And I mean, you mentioned kind of bifurcations, and I just wondered if you'd kind of explicitly tried to test for different attractors and then the distance to a bifurcation of a particular attractor or another in the data, or if that's something you're interested in combining with modeling in the future.
Yeah, no, absolutely. Thank you, Seth. So when it comes to criticality, I don't know about it. I know that it's becoming like a hot topic in the hippocampus per se. There's like a couple papers on bioarchive right now. They looked at it really carefully. In our case, maybe. But again, I still, get focused on this idea of like a phase transition for now. So we'll see if we move like further later. Of course, criticality, phase transition, all this is related, but without going into this big concept for now. What I wanted to say though is, yes, we have all this computational work now going on, in which we see these, representation emerge in systems. It's a very normative approach, meaning that we have like these recurrent neural nets without any inductive bias, like, nothing. We train them on different tasks. We train them to optimize certain objective function, and we see the kind of representation that emerge. Okay. So we've done it like with the model of the hippocampus, where we see that training these models to predict multi-step ahead in time was sufficient to see that the unit acquired place field, and that the network as a whole will become an attractor. Okay, which was like quite fascinating to us. So this paper is on bioarchive. Like Levenstein is the first author, and you even generate replay. Yeah, with like with the head direction, like signal, this you drive the system by noise then, and you will generate replay. Uh, I'm saying this because, of course, now what I want to see also is like whether we can use this approach to build a model now more specifically of the head direction system. And there's
Adrian, is
something,
this Daniel's work?
yeah, you know,
Oh,
you
cool.
know,
Yeah,
you
yeah,
know
great.
that.
We Daniel gave a talk to the lab last year when the
Oh
preprint came
yeah.
out, or
Oh, okay.
maybe
So
the year
you heard
before.
about
Yeah.
it. Yeah, yeah.
No,
Dan
we know
was
Daniel
was
well. It's great, beautiful work with Blake.
okay. Yeah, he was a postdoc in the lab, like he co-supervised with Blake Richards.
Okay. All
Now
right,
he
cool.
he
Small
has
world. I
his
was
own lab.
like, when you walk around a foreign city and you turn a corner and you're like, wait, I know this place.
Yeah. So yeah. Well,
Okay.
you know about it then. Cool.
No, it's
So
super beautiful work. We loved his talk. It was
thanks.
great.
Thanks. So yeah, now we're thinking of like building a model of what is it to get oriented before like thinking like we're going back, one system and say, you do head scan, you extract this information. So
Mm,
basically that stems from like, the behavior of any animals entering a new environment, like and think about yourself like stepping out of like the subway in like in this new subway station of you never visited the first you're a bit like even if it's a city like the first thing you do unconsciously is like you do a lot of head scan.
mm,
Okay, so in our view, like you're basically making predictions like I see this, I'm turning the head 90 degrees. What am I expecting to see? Oh, this is it. And by doing all these head scan, you're building a representation of space, which is basically a mapping between like this is how much I move, my head direction indicate that, and like what I
mm,
expect to see. And little by little, suddenly you build confidence into your prediction and you forget about it and you start navigating. So we want to see this like, also emerging from this normative, approach like this recurrent neural net. I can talk a long time about this because I think we're also very interested now in the circuit in the brain that will actually get this behavior, and
mm.
we think we have a good idea of like what is going on. So, yeah.
Oh, super cool.
We're working on it.
Thank you.
Okay. I have a bazillion more questions. Is there anyone else with a burning question? Maybe I can ask one more and then it's probably late for you. You also want to go home.
It's okay.
Can I ask you more an evolutionary question? Where did, there's animals like jellyfish that don't have probably this direction system. So where did this whole system start? Is it, with the event
Yeah,
of having a hit?
yeah, good, great question. So there is, as far as we can tell, there is a head direction system in all land animals. Fly have a beautiful head direction system. So it's like studied by a few group, like a small group of people, including like Vivek Jayaraman at the Janana Farm, for example, a beautiful system, which is a pure head direction system. Okay. Um, why I say land animals, because interestingly, if so we don't know much about like sea mammals like cetaceans, whales and dolphins, but one thing we know is that in these mammals these are outliers when you look at the size of the hippocampal formation uh, in relation to as a function of the size of the entire brain. So like their hippocampal formation usually is much smaller than what you predict from the size of the brain. So it is possible that actually when you're like a sea animal, you don't really navigate. And when you don't navigate, you don't need like a big hippocampal formation. So do you still have a head direction signal? I would be surprised you don't, because the system is so like, um, ubiquitous, and like they still have a vestibular system. So there might be, but like, what does it encode? Does it encode just the 2D? Does it encode 3D? Does it really help navigation? Does it do? And the reason why you don't have a hippocampus and like a shrunken, a system is also they don't have landmarks. Okay, so I've told you
Mm.
like all this, like how we extract landmarks from the external world, but like when you're at the sea, you don't have landmarks. Maybe you see like some faint, light from the sun, but they don't really orient themselves like the same way we think this is. So it's fascinating, hard to address as a question, but maybe one day we'll know more. But yes, in a system that exists like across all animals with a central nervous system, let's say, as far as we can tell. Yeah.
Wow, fantastic. You may have to come to Australia and run some, ocean study of some sort.
Yeah, yeah, that would be awesome. Yeah, yeah,
Awesome.
yeah.
Thank you so much.
No,
That
thank
was
you.
fantastic. Thank you for giving us a lot of your time at night. And hopefully we'll cross paths again sometime in the
Absolutely.
future. Thank you so
Absolutely.
much.
Thank you for having me and have a nice day,
You
everyone.
too. Bye bye.
Thanks. Great
Bye.
talk. Thanks.